Thank you. Thanks for the opportunity to get started early, too. Now I can talk for 20 minutes, right?
No.
As she mentioned, Mark Thomas, Department of Neuroscience. Glad that you all chose this session. So I was asked to put a few facts about my life up here. So this is what I came up with last night, some sort of embarrassing facts. But my dream job was not necessarily to be a researcher, for example. But this probably doesn't look like the face of a future rock star there, and so I had to think about a different career as I went on. I really wasn't terribly interested in science as a high schooler. Somewhat interested, but my science classes didn't really grab me. It wasn't until I got to college that I really understood, what science was all about. And part of that was that when you're doing high school science, a lot of times, at least in my area, you didn't get the chance to do new experiments that nobody had ever done before. So a lot of, when you're in labs, you're doing things that you're supposed to get the right answer, right? Because people have done these experiments thousands of times before. And so it wasn't until I got to college I realized, science is to learn new things about the world and not just repeat what others had done. So I was a little dense. That took me a while to get that. But once I got interested in that, I realized that the college I'd chosen really wasn't the place for me, and that I'd have more opportunities to study the brain, which is what I got interested in, if I moved to a different university. And so I transferred universities, and that was one challenge I had in trying to manage that process. But I got to my new university and it had lots of labs, and there were opportunities to get involved in research, and that's what I was able to do. And that kind of launched me into the next part of my life and career, which was to go to graduate school. That was another whole thing, trying to pick where I went to school, and there are many, many different choices. Ended up out in Los Angeles, which growing up here was a very different environment for me. But that was a great experience, and then that took me to several other places before I made my way back here. I feel lucky to have landed back in my home cities, and it's a great great research university here. Been here since 2003. And really, the thing that I got most interested in as a graduate student was how does the brain change with experience? And there are a lot of people studying that in the context of learning and how learning can go wrong in things like Alzheimer's disease or just normal aging processes, how learning starts to decline over time, the ability to store new memories. But I started to realize there were other situations that this might be a really interesting thing to study, and one of them was in drug addiction. So the idea is that with exposure to addictive drugs, this changes the brain in certain ways, and actually many, many different ways we're now finding. And some of those ways that the brain changes in some people produce patterns of behavior that can be really destructive and ultimately fatal. And so that's kind of the main challenge that has fueled my research for the past now about 20 years. And so we're working on this in models that I'm going to tell you about. So the main kind of question we've been interested in is identifying these brain changes that occur as a result of addictive drug exposure. I'm talking about things like cocaine, amphetamines, opioids, which you hear about a lot these days, heroin and oxycodone, things like that, as well as even as nicotine and alcohol. These things change the brain in ways that can be destructive. So we focused a lot of our attention on parts of the brain. This is a image of a human brain in a cross section down the middle. Parts of the brain that are involved in what we call reward. So there are natural rewards in our environment, food, the opportunity for sex, to procreate and keep the species going, as well as building social networks. So all these things are sort of sensed, and there's information stored about them in the brain in what we call reward circuits. Okay. So one of the parts of the reward circuit that hopefully you can see this little arrow here, that we've focused on is called the nucleus accumbens, this tiny little area here, which is has a powerful role in this reward circuit. And this, in order to study this and how addictive drugs affect this, we really need to do experiments on animals. Doing experiments on people is very difficult. Obviously, a lot of ethical issues involved. But when we do brain imaging in people, we don't have often a before and an after. And that's one of the big challenges. We do have that before and after in animal models like rats and mice, because we have our standard rats and mice that have grown up in the lab in healthy environments, but then we can administer or have them administer addictive drugs, and we can see before and after changes in the brains. So a lot of the work that has been done in our field has been done with that kind of approach. Okay? And these reward circuits have a lot of similarities between humans and rodents, which is kind of surprising, but these basic circuits are intact in these much smaller animals and much smaller brains. So I mentioned that the animals will take drugs themselves. If you give them the opportunity to press a lever to get an intravenous injection of cocaine, for example, they'll learn to do that pretty readily. Apparently it's rewarding, and so they'll press the lever, they get an infusion of drug, and this is a graph just showing you that over days of experience, they'll learn to press the lever many times to get a drug infusion. What we've been studying is how that changes the brain, and here we've got what we call a coronal section through rat brain, showing it in a living rat brain with fMRI, this is a magnetic resonance imaging technique, that we can see that there's stronger connections between portions of the brain, in this case prefrontal cortex and this nucleus accumbens that I showed you here. There's stronger connections after having had drug exposure, which is a little surprising. You think addictive drugs, they should be, damaging the brain. There should be holes in the brain when you look at it with imaging. Well, that's not really how it works. The addictive drugs can rewire the brain, changing connections between different regions, and thus changing cognition and behavior in some of the ways that can be destructive. So that's really what we're trying to figure out. But this is one area that we know is different. And so when we found this result, what we wanted to do was see how does that relate to the animal's behavior. And so we were studying a model for drug relapse. And this is resumption of drug seeking after an abstinence period. So many people who are taking chronic drugs can stop. The problem is to stay stopped over time. Very challenging for people. And so there are triggers in the environment, like cues that have been paired with drug in the past that will, 1 minute, okay. Whew. All right. Good thing I didn't do the dance routine. So that will resume drug taking. And this is a dangerous thing that happens in people that we want to try to understand better. There are ways that we can stimulate the brain that can change its connections. And this is what we, I'm going to skip over a couple points here. There are technical ways that we've learned about in neuroscience that we can use, using patterns actually of light stimulation to stimulate the brain, that can change these connections and change them back to what they looked like before there were any drugs present. So it's, in a sense, rewiring the brain back, in this very specific sense, back to what it was before this brain had any exposure to drugs. And we used a model here, as I mentioned before, that would, in the lab, be like relapse, okay? So we give, in this case, morphine again, which is an opioid, and now the animals normally experience that they go and seek the drug again, in a sense. They go to press the lever again to get the drug. But if we've done this brain stimulation, we can block that effect from happening. And so our goal is to take this from these experiments in rats and mice to try to figure out a way to do this in people that we might help to rewire some of those addiction-related things that may have happened in people who are trying to stay abstinent and clean. Okay, I better stop there. Thanks for your attention.
Any questions for Dr. Thomas? Time for one or two?
Yes.
How long do you think until this is actually available for human testing?
So the question is, how long might this be before this is available for human testing? There are some people who are trying to do this. It's going to be at least a few years, if not 10 years or more. There are a lot of steps in between this and making it available. But there are ways, and engineers are coming up with new ways all the time to stimulate the brain in very specific ways that we can do in animals that aren't yet available in people. Yeah. Yes? That's a very good question, and that's something we're really interested in. So we can focus in on this one connection and rewire it. How does that affect its relationship into all the other parts of the brain? We're studying that now. It's a difficult problem to get at, but that's why we're doing the brain imaging.
Okay, last
Yeah.
question.
What were some of the things that were tried besides light stimulation?
What were some of the, yeah, it's interesting. What were some of the things that were tried before light stimulation? So a lot of this work has been using drugs to try to interfere with the drug-induced plasticity. We don't think that's as useful as an approach probably as this brain stimulation. So that's where we're heading now. It's still relatively new. Thank you for your attention, everybody.
Okay, thank you.